It is my great pleasure to introduce our speaker tonight. Dr. Desjardin started off his mycological career as a young pup up in Del Norte County, collecting mushrooms with his parents, and maintained that passion for expanding his mushroom knowledge right on through his college career, including getting his master's degree at San Francisco State, studying under the late, great Harry Tiers. And so he got his master's degree there, and then got his PhD at the University of Tennessee, that exotic wonderland of mushrooms out east, studying with Dr. Ron Peterson, another wonderful mycologist. And after he attained his PhD and did a short postdoc, it was time for Harry to retire. So Dennis was able to step right in there to fill Harry's shoes, and he's done that magnificently. So he's currently full professor at San Francisco State and the curator of the Harry D. Tiers herbarium, which is has the largest collection of mushrooms west of Michigan. Michigan's the closest one that has anything rivaling San Francisco State's collection. His work has been based mostly in exotic, wonderful, beautiful places. I think the way that he works is he looks for a beautiful place that has been understudied with regard to mushrooms, writes a grant, and then gets to go there. So he did, for example, worked on mushrooms of Hawaii, mushrooms of Bali and Java. He currently has a mushroom research center in Thailand. He's worked in the South Pacific, as well as Principe and Sao Tome, which is off the coast of Western Africa, and in Brazil as well. And in working with Brazil, he's been focusing on bioluminescent mushrooms, those mushrooms that glow in the dark. And so that's been much of his recent work. I was fortunate enough to be in Dennis's lab from the mid-90s, I'm not going to tell you exactly what year, until 1999 when I attained my master's degree, basically in mushroom taxonomy under Dennis's tutelage, and I really appreciate that. So Dennis and his colleagues Mike Wood and Fred Stevens are working on, and very close to being finished with California mushrooms, a new field guide to the mushrooms of our state. So without further ado, Dennis Desjardin.
Need a little water after that. Thank you for having me back again. It's always great to be here. Geez, it's dark in here. I want to see the audience.
You want to turn those back on?
Just a little bit.
Okay.
They can see this nicely. No, you want it dark? All right, I don't need to see you.
Okay, yeah, turn it down.
You don't have a rheostat on there,
There's
right?
nothing.
Oh, then turn it off then. They don't need to see me.
Do you need a flashlight?
No, I got a screen here that's bright. And bright young graduate students here as well. It's great to be back here. I joined the Mycological Society of San Francisco in 1980. So it was like 33 years ago, and I've been lucky enough to be your scientific advisor since 1990 when Harry Tiers passed it on to me. And I want you to know this project is done. It's not almost done, as JR said. It's out of our hands now. It's at the publisher, and we hope you will have it in every one of your hands, and Christmas and birthdays and all the other gifts you can buy, sometime next fall. So what I want to do is I want to talk to you about our book, California Mushrooms. And if we had the lights on, you would see the two people I would like to introduce. Don't worry about it. We'll do it later.
Get the flashlight.
Yeah, the flashlight will do. It's like a spotlight. My two co-authors on this, both of whom have been president presidents of the Mycological Society of San Francisco. So this is really a local effort here. Mike Wood in the front row and Fred Stevens in the second row there. That's a on them. So it was a little over 2 years ago that I was contacted by Timber Press, and they asked me if I would write a field guide to California mushrooms. And I told them to call me back when I retired and I would consider it. They called me back a couple weeks later and kept asking me, and a couple weeks later and a couple weeks later, and I contacted Mike and Fred, who have been working with me for about the last 30 years. We've been collaborating on mushroom taxonomy. And we decided that, well, maybe now was the time to do it, even though I felt personally that I really didn't have the time to do it. It takes away from mainline research doing something like this. But their offer was pretty good. We had a pretty good contract, so we set upon writing this book. Now, the way we divvied up the effort on this is Fred Stevens likes gastromycetes, so he wrote the chapter on gastromycetes, and I wrote the rest of the book. And the photographs are mainly Mike Woods and Fred Stevens, and then for a few species we brought in some friends who have their photographs as well in here. And you see at the end some photo credits. So what I want to do is kind of tell you about this book, and to start with, here is the guy. You've heard about this guy. This is Dr. Harry Tiers. We've dedicated the book to Harry because Harry was a consummate teacher, all of our mentors, and a first-rate mycological scholar. He was a professor at San Francisco State from 1959 until he retired in 1989, and he was a scientific advisor of this club ever since he moved to the Bay Area from Texas, where he got his PhD. Actually, he got it in Michigan, but he lived in Texas. From 1960 to 1990, and that's what I looked like 30-some-odd years ago when I was a graduate student of Harry Tiers. So this book is really a product of what Harry always wanted to do and never quite got around to doing it. So we're trying to honor him by producing this product. So let me tell you about the book. Here is the contents for introductory materials. We tell you, first of all, what fungi are. There's an extensive section about the book. And one thing that has always kind of upset me about a lot of field guides and other books that are out there is they never tell you what decisions they made as to why they did the book, what the book is about, how they did the book, the decisions for what species were included, and that sort of stuff. So this is a chapter literally about the book, which gives you all that information about exactly what it is. And I'll just preface that by saying this book is California mushrooms. Every species that is in this book occurs in California. We don't have species that don't occur in California in the book. So it's not other West Coast species or just Oregon species. Every species in the book occurs in California. They also occur elsewhere, many of them do, so they have a wider distribution range, but we did not include anything that did not occur in the state. There's a chapter on nomenclature and taxonomy, which I'm going to hopefully not bore you with tonight. As an educator, I feel I have to both educate as well as entertain. So I want to educate you a little bit about nomenclature, what that is, and taxonomy, and some of the decisions one makes in coming up with the names that you put in a book like this. That follows right into morphology and phylogeny, so there's a chapter on that explaining the intricacies of both of those. I'm going to mention a little bit about that tonight as well. Then there's a chapter on the biology of mushrooms, which tells you about their life cycle and that sort of stuff, which leads right into what they're doing, why there are so many of them. A chapter on ecology. We go into a little bit talking about edible fungi and poisonous fungi, mentioning a number of the toxins that are involved. As far as edible fungi, that's my mom up there in the middle, and those are a box of porcini, some chanterelles, and some meadow mushrooms over there. What's to be insinuated is one in that complex that was described from the Berkeley campus by Kaufman back in 1924, and that's one of the most common things, or something close to that anyway, that we find in California. So a new name for that is incorrect. And a new name for other ones here. Insinuated. For insinuated, too. Yeah. Okay, because insinuated was described from the Berkeley campus. So this is the whole group here now. Geophylla is not in the book, but something in the group is in the book. Lots of hebelomas and this oddball, another bolete, another gilt bolete in the lower right there, Philoporus aerunicola. Yellowish brown, rusty brown, 39 species in this group, not including Cortinarius. In the upper left is one of our most toxic species. You've known this for years as Chemosity phalaris. In California, the correct name for the one that we have here is Foliatina rugosa. Beautiful little Bolbidia species, some Secotioid species, a little Galeropsis in the lower left there. And we actually added, I think, 5 or 6 Phaeocalybias to the book, including this species that was described from Mendocino County about 10 years ago as Benzokaufmannii. So we're trying to keep it to the restricted to California stuff. We have 25 species of Cortinarius. We could have put 100 or 200 in there if we knew them and had good photographs of them. But we do have a number. Cortinarius xanthodryophilus is a new species that Dimitar described just this year. It's found under oak trees. Down in the lower left is what you used to know as Thaxterogaster pingui. It's now called Cortinarius pinguis, and all kinds of the really viscid, gooey Phlegmaceum and Mixaceum groups of Cortinarius. So quite a few Cortinarius, more than pretty much any other field guide. And Agaricus as well. Fred likes Agaricus, so we've got 23 species of Agaricus in the book as well. In the upper left is what you've known as Agaricus arvensis, typical horse pasture mushroom for years. Agaricus arvensis is not here. The correct name for our Agaricus arvensis is Catturatus. In the upper right is something you may have called Agaricus praeclarisquamosus for years. There are 2 or 3 of them, if you've said that word at all, right? It's one of these phenol-odored, really awful smelling, awful tasting yellow stainers. And this is a group of these 3 species up there. And Rick Kerrigan has done the work on this. He has a North American monograph of Agaricus that's in press right now, and it should be out soon. He described this as Dyrgarfensis. And then what we had in the mountains that we've been calling Agaricus sylvicola for years is now called Agaricus moronii, named after some moron. And on the lower right is a little rare one that you find in the grasslands on occasion. This is Agaricus micromegathus. Some wonderful coprenoid fungi and beautiful photographs of these.
One moment.
Yes.
What does micromegathus mean?
I have no idea.
That sounds like
Something small and something big at the same time. How about that? Me and my ego. Sorry. Coprenoid fungi. We've got 17 different coprenoid fungi in here. I'm just showing you some of the smaller and more beautiful and odder ones. Some Coprinella species, Coprinopsis. The one in the middle at the bottom we have in quotation marks because it falls in the middle of Coprinopsis and Coprinellus and the phylogenetic trees. It's all by itself, and it's not a Coprinus, a Coprinellus, or a Coprinopsis, but that name is available for it. And it's Ephemeroides. This lasts just a few hours. Really a beautiful fungus. And this beautiful parasol on the right, now in the genus Parasola, Parasola lactea. We have a chapter in the dark-spored ones, the Lacrymaria, Paniolus, and Sapphirella with 10 species. And there they are. Pretty typical ones that you've known before, if you've studied these at all. We have a chapter on Hyphiloma, Psilocybe, Stropharia, and allied, so this would be basically Strophariaceae, 11 species. You should know that all of the Psilocybe species that are not hallucinogenic have been moved out of the genus Psilocybe into the genus Diconica. And the hallucinogenic ones all hold together very nicely in a phylogenetic tree, and the non-hallucinogenic ones hold together, and they're quite distant from each other, believe it or not. So this is now Diconica subdicida. We have some beautiful Secotioid fungi. This used to be Weraroa cuculata. It's now Loratiomyces cuculatus. Down there on the left is something you probably called Stropharia summiglobata in the past. That in the phylogenetic trees is all by itself, so they erected a new genus just this year called Protostropharia summiglobata. And then an odd one, again, in grasslands, a very small one that you may or may not have seen before, Stropharia coronilla. So again, some of the oddballs. Only 5 in this group of Crotogonthus and Gomphidius. You know pretty much all of these. What you used to call Crotogonthus rudulans is the correct name for that, or the currently accepted name is Crotogonthus ocraceus. On the lower left was another Secotioid. One is now called Gonthus albidipes. I learned it as Browniella nancii. So these names are just changing, unfortunately. and the little beautiful reddish one down on the lower right, which is very common in Mendocino County northward, Gomphidius subroseus. We have 44 boletes. Lots of boletes. Lots of boletes. And some, I'm just showing you the odd ones here, just show you some of the name changes. In the upper left, you probably, those of you who've collected these things, knew this is Talopolis pseudoscaper. You will find it in all the books as Talopolis pseudoscaper. People recently have said, well, that it doesn't really belong to the genus Talopolis, it belongs to the genus Porphyrelis. So that was kind of one of those taxonomic decisions to move it into the different genus Porphyrelis. And they said that pseudoscaper was a synonym of a European species, Porphyrelis porphyrosporus. So in one field guide, you're going to see this called Porphyrelis porphyrosporus. The molecular work that's been done by Roy Halling on material from California shows that the California species is different from the European Porphyrosporus, which was described from, hence it needs another name. Well, as it turns out, a number of years ago, there was a species described from Oregon as Talopolis olivaceobrunneus, which is exactly the same thing as what we have here in California. So this is a combination that needs to be made. It's a provisional combination in our book. Olivaceobrunneus is the better name for this taxon, and it should be moved into the genus Porphyrelis. So that's one of those names that is still yet to be formalized. Lots of Swillas, about a dozen Swillas in the book. This little tiny guy, Megapyrinus. Another one of these name changes in the lower left is something called Boletus orivillus. Harry Tiers described this. It's one of the few Boletus that is not mycorrhizal. It's actually a saprotroph. And it actually belongs to a genus that has been recognized in the last decade or so, Buckwaldo Boletus. So I want Fred to take a picture of this kind of hiding in the woods, and we can have under there, where's Buckwaldo? And on the lower right is what you've known as Boletus dryophilus. It now belongs to this new genus that was erected just in the last 5 years or so, Xerocomelus. So this is now called Xerocomelus dryophilus. These are all pretty much based on molecular characters. A lot of polypores, more than I would like, because people who know me know I hate polypores. But this time of year, you can collect them and find them because there's nothing fleshy out there. There's our common sulfur shelf on conifers, Ledoporus conifericola. You know by now that there is no Ledoporus sulphureus on the west coast. There's Ledoporus conifericola, there's Ledoporus gilbertsonii, which is on hardwoods. We have a number of Phylinus in the book. This one grows on plum trees. On the lower left, things you used to call Pyromyces are now in the genus Postea. And these beautiful pink ones like Fomitopsis. So lots of polypores as well. 17 spine fungi. Some of the typical ones are Hericium aviatus in the upper left. Oddball ones like these on the right. They're not really spines. They're actually straws. They're single tubes. And the spores are produced on the inside of them, not on the outside of them like spines. But we couldn't find any other place in the book to put something as odd as that. They kind of look like spines, so they're included with the spine fungi. Then we have the beautiful little yellow ones, which are just single little spines or little clusters of spines that hang off of wood, genus Mucronella. And then your typical little hedgehog fungus on the lower right, Hydnum umbilicatum, with the little umbilicus in the center of it. Another group that you might not be familiar with, the Telephoroid and Stereoid, they have the spore-producing surface smooth, without teeth or pores or gills or anything like that, smooth or wrinkled. So we have things in here like Thellephoras and Steriums and this beautiful Voludiceps, and we couldn't figure out where else to put Sparacis. Now Sparacis has a smooth spore-producing surface. It actually produces spores on both sides of those petal-like structures called flabelli, but it's actually a polypore. Sparacis is in the Polyporales, in its own family, but it's a polypore. But since it has that type of a body form, that Telephoroid stereoid body form, we included it in this chapter. And then club and corals, 34 of them. Lots of Ramarias, lots of beautiful Ramarias in here. A couple examples of clubs. In here, here's where we kind of stepped out of the boundaries of most field guides. We included some club-shaped ascomycetes in the same chapter as club-shaped basidiomycetes. And they're designated as that and separated in the chapter. But the one on the lower left is actually an ascomycete, and the one in the upper left, the Clavaria delphis, is a basidiomycete. Lots of the coral fungi on the right. In the upper right is something that you may have known as Clavulina or Ramaria myceliosa. Some people say that it should be called Phaeoclavulina curta, but curta was described from Europe. myceliosa was described from Stanford. It's a West Coast species, so I'm convinced that what we have here should be called myceliosa. And that's another name that has not yet been formally transferred to Theoclavulina. And lots of the beautiful little bromeliads as well. Gastromycetes, Fred's chapter, 37 species in here. Things like the beautiful Calvacia sculpta, lots of earth stars, Diastrum floriforme, lots of bird's nest fungi in the lower left, and a bunch of stinkhorns in the lower right, and all the different kinds of stinkhorns that we have in California are in the book. So this is a pretty thorough representation of California gastromycetes, probably 95% of them, if not more, in the book. And then the crust fungi that nobody collects, but we had to throw them in. So this is basically you roll over a log and you see some little crusty fungus-looking thing on the lower side. That's a crust fungus. And they are really, really diverse. There are hundreds of them in California. We included 12 in the book because that's all we had photographs of. From the beautiful blue ones and the orange ones. Serpula hemantioides down on the lower right. That's related to the dry rot fungus. But this is one of our native species that occurs here. And the one on the left is actually a jelly fungus, Sebacina. Not only is it a jelly crust fungus, it's also mycorrhizal, which is very strange. And then we have jelly fungi, 10 of them, and you see the typical jelly fungi here. A really oddball one in the lower left, now called Mixarium nucleata. It's got a little core, a white core inside of it, and then it's surrounded by a clear gelatinous ball. Looks like a little fish egg. And then we've got 13 different morels, false morels, and elfin saddles. So the common one that you get in the coastal areas here, in horticultural areas, disturbed habitats, the blonde representative is Morchella rufobrunia. Some of that was brought into your fair. I had Brendan bring some in. It came up on campus a few weeks ago, I guess last month, and he brought some in for you guys to see it at a meeting last month. Gyromitras, Helvelis. On the lower left there, just so you know what's going on, the morels in North America have gone through to molecular turmoil. There have been a number of people doing some work on this, sequencing a lot of the morels, the burn morels, the native morels, all the morels that you can imagine. And there was a paper out last year on the molecular phylogeny of North American morels, and they came up with a whole bunch of names. And the most common black burn morel that we have in California matches the clade number 6 or clade number 7 in the tree. And since the authors of the paper weren't nearly as creative as I was in naming something like Spongiforma squarpansii or something like that, they decided to name these morels after the clade name. And since it belongs to a clade called the Morchella elata clade, they came up with number 6 is sextimolata and number 7 is septimolata. So those two are completely indistinguishable morphological. You cannot tell them. But if you happen to have a pocket sequencer on you and you sequence the ITS region when you're in the field and then you run a phylogenetic tree, you can tell whether or not you've collected sectilata or septimolata. So our book just calls them Morchella 13. Just kidding. But Mike wanted us to do that. Cup fungi, just about done here. Cup fungi, 35 different species of cup fungi, and these fascinate everyone. Beautiful ascomycetes here. From every color you can imagine. The one on the upper right used to be called Sarcosoma mexicana. The correct name for Sarcosoma mexicana now is Ernula pediniana. And even worse than that, just this year, some Europeans were looking at this beautiful thing that we called Plectenia nanfeltii for years. It's a little martini-shaped thing in the lower left there that's black. It'll hold about 1/4 of an ounce of whiskey. My graduate students have proven that. And we've called that Plectenia nanfeltii for years and years and years. They did the phylogenetic trees of the Plectenias and the Pseudoplectenias and all those things and found out that it forms its own little clade in the tree. So they erected a new genus around it. They called it Donadinia. And an older name for it is Nigrella. So it's now called Donadinia nigrella. And the one on the lower right is named after hairy tears. It's a new genus that my former student and postdoc and Brian Perry, many of you know him, I think he spoke here recently. This is something that we've been collecting for years up in the Sierra Nevada, and they felt they wanted to honor Harry, who collected the type specimen. So they decided to honor after hairy tears. It has hairs all along the outside of it, which are called kiti. But it actually translates to hairy tears, which is really cool as well. So a nice little alliteration there. And then we have truffles in the book as well. We've got 17 different species of underground fungi, truffles and false truffles, true truffles like the genus Tuber candidum, false truffles like the rhizopogon, sedecula down there, a very odd fungus there, and underground stinkhorns like Trapea darkeri. So you'd never do this alone. We called in some consultation for some friends who, when we asked them what species of Lepiode do you think we should include in the book, Elsa came up and said, well, here, how about these? Other people in there, Rick Kerrigan helped us a lot with Agaricus. We asked him, these are the names that we have. He says, well, I have a new monograph now based on molecular characters. And so he sent us unpublished data that helps out a lot. David Largent, I forgot to put his name up here. Is it there? Oh, there it is. David Largent, we asked him about pink sport stuff. He gave us a whole list of pink sport stuff. We asked people from the south, Steve Penkel, what grows down there. He gave us their list. So many of these people have helped us decide which species we should include in the book, and a number of them have also provided photographs. Down there at the bottom of the photo credits, so you can see how many photos Mike has in the book and how many Fred has. And other people that we've called in specific photos. Dimitar was very helpful with some Cortinarius photos for us. And then a smattering of other people gave us. Taylor Lockwood, of course, we can't do anything without having at least a few of Taylor's wonderful photographs in the book. So he's donated the photos as well. So we definitely want to give credit to all the people who have helped us along the way, either through consulting or with critical comments about some taxonomy, or who have provided the photos. So here's where we are now. The book is at the publisher. They are going to make a hardcover out of it. It's a large format, 7 3/8 inches wide by 10 3/8 inches tall. It's going to be approximately 900 pages, which is about 2 inches thick. As I said, it's going to have 650 comprehensive descriptions and color photographs, treating 1,100 species. And it will be available, they tell us, in fall of 2014. It's out of our hands now. They're formatting it. They've given us some sample pages to look at. We've rejected a number of them. We've accepted a final format. And we think it's going to look beautiful, and we think you're going to like it. And you can always hold your door open with it if you don't want to use it. And I'll leave it at that. Thank you. Say one more thing. All these descriptions are based only on the morphology that is found in California populations. So all the descriptions, all the microscopic descriptions are ours, from data that we've collected over the last 30 years or so to generate these descriptions. And they pertain specifically to California populations. Now, you find these species maybe in Washington or some other states, or maybe even some of these species that occur back east. They may not match our full descriptions and some characters, because we're talking about, the book is called California Mushrooms after all. So I just wanted you to know that as well. And I'll take any questions if you are not asleep yet. Norm, and then I'll go to the back right there.
Are you going to put an online errata so we can...
That's a good question. Mike has already set up a website for online errata. So already some of the name changes, as soon as the book is published, it'll go active with the errata on it and some potential name changes.
I haven't put the errata on there yet, because right now, it's not any use right now.
Right. Yeah, but there will be a website for name changes or any corrections and things like that.
CaliforniaMushrooms.us and see a couple sample descriptions and a few of the photographs.
Yeah, there's a website for it already. You just Google California mushrooms, you'll get to it. Yes.
Do you have any information about their tree relationships, if there are any, if they're pathogenic, or whether they're interacting or not?
Yes, that information is pretty much in there, in the book. Each chapter has an introduction to the chapter, which talks a little bit about the ecology of the various genera that are included in the chapter. So it's talking about whether they're saprotrophic or mycorrhizal and pathogenic. In the commentaries, if it's pertinent to understanding the species, the commentaries will mention something about that as well. Yeah, and then there's habitat information for every species, so it tells you where to find those. So you know, if you want to collect Boletus edulis, it doesn't say go look under oaks. Now we could send you to the wrong place, but we're trying to send you to the right place. Sure, behind Danny there. Danny, you can ask me questions.
With all the new names, are the old discredited names referenced?
Yes, so what we've done is we have a list of synonymy for every species as well, if it's pertinent. So any of these species that are known by some other name by most of you and are in any of the other field guides by that name, it's listed there either as a synonym or as a misapplied name. So many of these are listed. It's all very specific there. And those will all be indexed as well. So if you can't find your favorite fungus because it's got a new name, go to the index, look for it, it'll send you right to that page. Yes?
How many endemic mushrooms are there in California?
I have no idea. Lots. How about lots? I've never counted up the ones that we know. So the problem with they're endemic only as long as you haven't found them somewhere else. And our knowledge of the distribution of fungi is just really, really weak because not enough people collect them. It's not like a lot of plants which are out there for long periods of time where you can see them, right? Fungi have sex when they feel like it, not when we want them to. Like I want them to have sex right now. So I have mushrooms for my class. They're just saying, forget it, it's too warm. The mycelium is sunbathing, you know, they don't want to have the sex. So we don't know. We got to wait for the mushrooms to come up, and then someone will report it. And then if somebody finds it in Oregon, it's no longer a California endemic. And so you have to go with the habitat. So many of these are also substrate specific. So for instance, we have one in the book called Microamphilia ruticola. It only grows on the scale bark of arbutus, madrones. And wherever you find madrones, you will find it. Now most madrones are in California, but madrones go all the way up into Oregon as well in southern Washington. So you will find it up there as well. It's described from here, thought of as a California endemic, but if you look hard enough, you're probably going to find it wherever madrones go. And that can be said for a lot of these relationships with the fungi. The habitats, so they're pretty much restricted to habitats. Yeah.
Is it, do your different chapters have dichotomous keys as a typical field guide?
Every chapter starts with the key to the species included in that chapter. Dichotomous key. And we hope you use the dichotomous keys. The publisher was publishing them single column, little font, and that was one of the corrections. Now it's full page, big font. We want you to use the keys to get to the species. Absolutely.
As a field guide, does it come with a carrier?
Come with a, it's going to come with wheels. Little pop-out wheels in the back. You can drag it behind you like a suitcase. All right. Yeah. Come on.
Did you include anything about like medicinal uses in the edible section?
Yeah, on occasion we have. For the species that are commonly collected that obviously have some medicinal use. In the commentaries for that species, we mentioned that this has been used for this and this sort of stuff. This could be a mycomedicinal, etc. But it's not a lot of information in there on every species that is in the book that may have been used medicinally or not. Yeah.
Do you also have information on their use as dyes?
A couple species that are in the book that are commonly used as dyes, we mentioned it in the commentaries. That this is also used by people to color fabric or textiles. Yeah. A couple more questions and then I'll let you guys go. Yeah.
I have a question about the mushroom formerly known as Boletus orville.
As Boletus orvillus, yes.
Two questions. Is that one mycorrhizal or is it saprobic?
It's, as far as we know, it's saprobic.
Really?
It's found typically around dead trees. It belongs to the genus based on all of its morphology and stuff, Buckwalda boletus, which is the group that holds most of the saprotrophic boletes. The genus Phlebopus is another one that's saprotrophic, so there are two saprotrophic genera of boletes. And Buckwalda boletus it's got little ellipsoid spores. It's not like a typical bolete spore. It's quite different.
And the other question is, horribilis, is that named after gold fingers or is it named after Orville?
It's named after Orville. It was collected in Orville, California, the type specimen.
But does that translate properly as gold finger?
You'd have to ask James Vaughn. getting late. Yeah, one more question, then I'll go. Yeah, you.
Is it just Basidios and Ascos, or do you have anything else in there?
We have one zygote in there, only because it occurs on Mycena. Spinella, you know, that you get little Mycenas and you have those big hairs that stick out all over the place with little black balls at the end of it. That's in the chapter on Mycenas. Otherwise, they're just Ascomycetes and Basidiomycetes. They're really the only ones that form something that we would call a mushroom. If you could think of anything else that you would call a mushroom that's not there, I mean, you know, the zygos and the glomerules and those sorts of things really don't form mushrooms. So it's basically just these. No rust or smuts, no slime molds. That's the next time. What I wanted to do, I told the publishers they were crazy to do this. I mean, why publish a big hardcover color book? I want an app for my iPad. So I don't want to be restricted to 240,000 words. I talk way more than that. I ask my students in a single lecture. So with an app, you aren't restricted to words. Pixels are free. Bytes are free. You know, so I'm hoping that eventually, in a few years down the line, we can convert this into an app where you have 10 or 15 pictures for each species, and then a link to the PDF where the species was described, and all the other kind of cool stuff you can do with an app. I mean, that's where this is 2013. Come on, but the book publishers are still locked into this hardcover stuff first, then softcover, then an electronic copy of the hardcover, right? And then maybe we'll think about something new age, like, you know, a smartphone or something. It's crazy. Thank you very much. We'll see you soon.